Okay, so I'm gonna first start out with light reactions, which happen in the dichloroid space. At the top is the stroma, at the bottom is the lumen, and then there's the dichloroid membrane. And so a photon of light excites electrons. Oxygen, the oxygen evolving complex is in the lumen in photostem 2 complex. Water is splitting the enzyme, splitting the H2O into O2 and H. As you can see there, the O2 is going to go into the PQ, and then that changes to the PQH, and that is going round and around. So the PQ changes to the PQH, going into the cytochrome B2 complex, which is actively pumping H ions into the dichloroid space. Here it's also picking up electrons and sending it to the glycan, then sending it into photostem 1, which is also picking up a photon, exciting an electron. Plastoglycan makes way through photostem 1 into the ferredoxin, changing the ferredoxin into ferredoxin NADP reductase, which picks up NADP and changes it into NADPH. And then the H plus ions go all the way to the ATP synthase, which is right here, which is basically the goal of light reactions, to make ATP. So next I'm going to go into the Calvin cycle. So the Calvin cycle starts with the ribulose 0.15 biphosphate and picks up the CO2. It enters with the ribisco enzyme, changing it to 3-phosphoglycerate, then picks up ATP, changing it to ADP and adding the last phosphate group, making it a 1,3 biphosphoglycerate into glyceraldehyde 3-phosphate. One of those leaves and changes to sugars, fats, and amino acids. Example of that would be glucose. The rest goes up to let go of 1-phosphate. The ATP comes in and donates a phosphate group back to and then goes back to ADP again, and then all the way back to ribulose 0.15 biphosphate.